Okay, I'm going to go ahead and get it started. Thanks everyone for joining us today. The July installment of our statewide lab advisory meeting. Just as a reminder, if you could keep yourself muted, feel free to ask questions or something, but just so we can cut out the background noise. Or if you have questions before, after, during, you can always email us at nphlab@unmc.edu. So we'll go ahead and get started. The meeting is being recorded. It serves as the minutes. It's also how we kind of keep attendance. So if you haven't, put your name and facility in the chat. And we'll move on to new staff introductions. I don't know if we had any new hires in the last month. We'll open it up. Okay. Sound like we got any new people. Okay, so I'm going to move on to laboratory topics. The first one, the media shipment changes. A lot of you may or may not know, every 6 months we send motility media to a handful of labs around the state for your rule out and refer procedures. Something we've been talking about for a long time is discontinuing the motility media shipments. The cost incurred and the man hours required to kind of put all that together is starting to, the lemonade is not worth the lemons, as one might say. So I think September is going to be our last motility media shipment. But that being said, for rule out and refers for Bacillus anthracis, we're not going to require the motility result. You know, if the morphology of the gram stain in the colony is consistent with Bacillus, if the patient history is consistent, we're just going to go ahead and collect those for rule out testing. You can email myself for that NPHL lab email with questions or concerns. I don't know if Pete's on, if he has anything to add to that. It's something that's kind of been in the works for a little while now.
No, can you hear me?
Yeah, we can hear you.
Yeah, I think that it's just a smart move for us. It's a huge headache for us to keep track of. And we realize that we may end up getting more of these for rule out to the public health lab, but I think it's, you know, something that we can deal with, and if it becomes a real burden for us at the public health lab, then we will reevaluate this. But in the meantime, we're going to give it a shot. So we'll see how it goes. Yeah.
And we are we're evaluating some of the other media we send out to the urea, the indole, the oxidase. We're kind of taking a look at all that and laboratory capabilities too. You might have noticed some surveys coming about what kind of reagents you had on hand in the lab and what testing capabilities are around. So we're kind of gathering all that information and assessing our rule out procedures. So, but feel free to get a hold of me or Becky with questions or concerns. We can talk about it more. So moving on here, something we also talked about here at NPHL was doing short little informational presentations at these meetings, kind of pulling the curtain back about what goes on here at NPHL. And so this month in our first little mini learning session, Jeff Hammock and Becky Bonacci were kind enough to volunteer to talk about mosquito testing in Nebraska, kind of what the processes are and what happens behind the scenes. So I'm going to let Becky take over here. Becky, are you ready to go?
Yes, hello. Jeff, are you on?
Yep, I am here.
Okay, you want me to just share this PDF, or do you have slides that you'd like to
I can share my screen if that works. Either way, I got
Yeah.
slides here or so.
Yeah, if you'd like to share the slides, it looks a little bit better than this PDF.
Okay.
Oh, yeah.
All right, let me go to share screen here. All right, hopefully everyone can see that.
Yep.
All right, we still good?
Yeah.
Okay, perfect. All right. Well, I guess good afternoon, everyone. For those that don't know me, I'm Jeff Hammock. I'm the Vector-Borne Disease Surveillance Coordinator here with Nebraska DHHS. And yeah, we'll just talk a little bit about our mosquito surveillance program here in the States. So the reason we do mosquito surveillance is because there's a couple reasons. Well, if we just use human disease surveillance by itself to predict West Nile outbreaks, it's typically insufficient, and that's because human West Nile virus case reports they lag several weeks behind when transmission from mosquitoes that initiate the infections actually happen. And so we're kind of getting we're not getting what's the true risk of what's going on. We're kind of seeing the aftermath of after the risk has already passed. And so the purpose of surveillance is to help monitor that infection prevalence in mosquitoes and help provide that early warning to the public of West Nile virus circulation. And typically we can detect human risk about 2 to 4 weeks in advance of human cases starting to get reported. And additionally, we can help create mosquito distribution maps in the state. We can help us focus intervention measures. So that might be mosquito control, that could be public health messaging. And we can also use it to detect invasive mosquito species. And we collect mosquitoes from across the state. This is a really good partnership we have with our local public health departments across the state. Each local public health department has at least one city or county that is participating in our mosquito surveillance program. And the way we have it set up is that we have half the counties trap on an even MMWR week, and then the other half trap on an odd MMWR week. And that's just because we would love to trap every county weekly. We just don't have the funding to do that, nor the space at our lab to really keep all those mosquito samples all at once. So that's why we divide it up across the even and odd weeks. So in our surveillance, we utilize what's called a CDC light trap. This is kind of our bread and butter. It's a pretty simple contraption. It uses a small light and a fan. And then, yes.
Are the slides supposed to be moving forward?
Are they not going forward? Okay,
No.
All right, here we go. Sorry about that, everyone.
That's right.
Did you want to put it in presenter mode?
Well, that's what I thought I did. It's on presenter mode on my side, but apparently.
Maybe it's on the wrong screen. Oh, wait, you only have one screen.
Yeah, so that's right. We'll just keep with this here for now. So, so we utilize the CDC light trap. It's pretty, like I said, a pretty simple contraption. Uses a small light and fan and a dry ice that we put in this little thermos here as attractants. That brings the mosquitoes that are in the area up close to that trap. And then once they get close enough, the fan kind of sucks them down into this little collection net down here and holds them there until they're picked up. Typically, these are set overnight. About 4 to 6 p.m. is when they're typically set out. We usually want to set them up near known mosquito breeding sites, particularly in areas where there's some tree cover and tall brush. And that's because mosquitoes will typically, during the kind of hot part of the day, hide out in these tall brush and treed areas just to keep out of that hot sun. And then again, as I said, they're set overnight, so they're collected in the morning, usually around 8 a.m. And this trap type it's really good at collecting a very good number of different species and different types of mosquitoes, and it's really efficient at collecting Culex mosquitoes, which we're of most interest in because those that species of mosquitoes are the primary West Nile vectors in the state. And then here, it's just kind of a sampling of the different number of species that we can collect from these types of traps. This is by far and away not all the types of mosquitoes we collect. There's some years where we can collect 30 to 40 different species from our traps, just depending on kind of on the environmental conditions. But this is kind of a smattering of some of the common ones we typically will see in our traps. And then with once the collections are picked up by the local health departments, those are then sent to our entomology lab here in Lincoln. And once we get those samples, we go through them and identify and count all the mosquitoes that are in there. And then what we do is we keep the Culex species out of the samples. We will pull them out and we'll save those for pathogen testing. And what we do is we'll pool the Culex mosquitoes for testing. And when we pool them, what I mean is we'll put up to 50 mosquitoes of the same species, collected from the same date and site, and we'll place those into a tube. These pooled samples are then sent to the Nebraska Public Health Lab, where they're tested for 3 endemic arboviruses. West Nile virus, our most common one, St. Louis encephalitis virus, which we do still detect occasional positives, and then Western equine encephalitis virus, which we haven't detected positives in quite a while, but it is still an endemic virus we kind of want to keep an eye on. And so that's kind of the field side of the mosquito surveillance world. And so I'll turn it back over to Becky and let her then talk about the testing of the mosquito pools once they get to MPHL.
All right. Thank you, Jeff. Okay, can you see my slideshow?
Yep.
Okay, good. All right, and before I get started, thank you to Jeff, but also thank you to Amanda Bartling. She is the biology section supervisor over here, and she's the one that took all the pictures for me, and she walked me through the process and answered all my multitude of questions so that I could present this to you. And just as Jeff said, those mosquitoes are pooled, and this is exactly what they look like. The one on the right has 50 mosquitoes in it, and the one on the left has only 1, which is obviously our minimum. Now, our testing essentially is 3 different stages. The first stage is processing the sample. We have to do something to get that RNA out of there. The second stage is extraction, extracting the RNA out of that sample. And then finally, we're going to put it through a PCR process to amplify that and get our result. Okay, so this right here is the beginning of our processing. We've added some phosphate buffered saline to it. From anywhere 150 microliters to 1,500 microliters. And you could see at the bottom of that, there is a bead at each one of those. And the reason for that is we have to beat the stuffing out of them, to be quite simple. You can see at the top picture right there where the tech has taken all the pools and organized them evenly. And on the right, those racks have been placed in our machine, and that is where the pools are beaten. And on the left is the end result. And you can see quite clearly how cloudy it is. Now, we can't just test from that. There's too much debris in there. We have to put it through a centrifugation. As you can see on the right, all of our pools are in that centrifuge. And what that does is it takes all of that debris and pellets it at the bottom of the tube. And this is what we're left with. Now we are going to take the supernate off of those, and that is what we're going to be testing. That is where the RNA is going to be located in this specimen. And as far as time goes, it kind of varies on how many pools we have. But generally speaking, for every 60 pools, it takes about an hour to process these specimens. Now, the next stage is extraction. Now, on the top of your screen, you can see where all of the reagents and all of the pipettes are and all of that. And that's what they have to get ready for, along with at the bottom, all of the pools set up, getting ready to put in their slides. And this is our extractor. If you can see the racks there inside the extractor, it takes about 30 minutes for the extraction and the purification take place for the RNA out of that specimen. And that's what we're going to use for our PCR process. And that's our final stage is the PCR. And you can see on these slides on the left, she's getting ready to add master mix to the plate. And on the right, she's getting ready to add all of the extracted samples onto the plate. And this is our machine. This is the ABI 7500. And from between batch time is about 10 to 15 minutes to getting it prepared to put on the machine. And then PCR time, it takes 1 hour and 45 minutes. So about 2 hours once it's the PCR stage starts. And this is what we're left with. These are all the amplification curves from the pools. Now, what's positive and what's negative? Well, if you could see all the curves up toward the top, there's 1, 2, 3, 4, 5, 6 curves that we would consider positive. Now, on each run, we do a positive and a negative for the St. Louis encephalitis and the Western equine encephalitis, as well as the West Nile virus. So we'll see the positive controls in there as well as the pools themselves. So we have 3 positives in all of this pool. Now, this is where our testing stops. We do have to report. And I would like to note that when we do reporting, the testing individual does put those results in, but then a second person comes along and they do verify that before it goes to the state. And that's when it gets to the state, that's when it goes to Jeff's group and he determines risk across Nebraska. I don't know if Pete wants to mention anything about this. He did tell me right before this meeting started that we don't just finish with there. We do take our pools and we do send them up to the College of Public Health so that we could do further genome testing. Did you want to say anything about that, Pete?
No, you mentioned it, that we do have another need for these positive pools. That we're looking at genomic surveillance of West Nile in Nebraska and actually going back a number of years and looking at how it's spread. And Jeff, I think you're involved in this discussion as well, but we've got a researcher here on campus who's willing to take these positive pools and actually sequence the West Nile virus and do sequence comparisons. So I think it's a pretty neat study. I think it's somewhat unique in the United States that we're doing this. And hopefully we can maybe get South Dakota involved as well, because North Dakota, South Dakota, Nebraska are the hot spots for West Nile virus. So it'll be a neat study. I think we would consider it a pilot study, and hopefully we can get funding from the CDC, et cetera, to help us support this. But anything you want to add, Jeff? I think it's pretty much a new program that we put in place.
No, I think you hit everything there, Pete. Yeah, it's something new we're just looking into, and yeah, I'm kind of interested to see what comes out of this this new project. But yeah, and as Becky mentioned, yeah, we utilize, once that data comes through, we utilize it. And right now, another project we're working on is an experimental prediction model that was developed by South Dakota, their state health department and some of their South Dakota state researchers. And we're kind of in an experimental phase right now, kind of looking at how that will work with Nebraska data, both historic and current data. So we're looking at that, and then hopefully, if it looks good and actually does some pretty good, predictions, maybe something we'll implement here in the future.
All right.
That's good, Jeff. I'm not sure people recognize this, but Nebraska, South Dakota has the highest incident rate of West Nile virus in the United States, and I think Nebraska's second or third. So we're hot when it comes to West Nile infections, positive mosquitoes here in the Midwest. So it's something that we can do that, is unique to our environment.
Okay. And that's it for the end of my presentation. Again, I want to thank Amanda Bartling and Jeff, and I think that's all I have.
Thanks, Becky and Jeff. I think that's very helpful to understand what's going on with West Nile.
Yeah, thanks a lot. So, like I said, I think every month we're going to try to put together just a little educational spiel. We'll come up with a different topic, and the HHS folks, we will probably be reaching out also to you, just like we did Jeff for this one, for additional information, and we'll try to put together little educational experiences for everyone every month.
There was a question in the chat about malaria testing, plasmodium testing of mosquitoes. Jeff, can you share about that? Because we know that that's percolating down in the south right now.
Yeah, no, we don't do any testing for malaria. We know it's a low risk here. And, it's one of those things, we get these occasional outbreaks about every 10, 15, 20 years, but there is no program in place for that. There's just, we don't have funding that would pay for that. I think it would certainly be interesting as a research project to do, mosquito surveillance around some of these, bigger airports where there's a lot of travel from endemic areas with malaria, just to see how many positive mosquitoes they might pick up that maybe we don't know about, because I know certainly there are a lot of travelers that come back that are infected with malaria. And, I wouldn't be surprised if mosquitoes are picking this up more often than what we, would we think of or expect. But yeah, I think it'd be certainly interesting research project, but yeah, right now there's just no funding for it. Unless there's other states maybe that have a little bit more public health infrastructure built in that can do that. But as far as we our funding comes from CDC, and they don't have funding for that right now.
Say, Jeff, do we see the Aedes mosquito vector in Nebraska?
Oh, you mean Anopheles mosquito? Yes, we actually we do have several species of Anopheles mosquito, and a lot of people don't realize this. Malaria used to be endemic in the state of Nebraska back in the late 1800s through I think even the early 1900s. It was actually endemic in Nebraska, and we still have the primary malaria mosquito that was responsible for a lot of the transmission in the U.S. It's Anopheles quadrimaculatus. We actually have that species still that's circulates around the state, and then we have several other species that have been shown in lab studies to be able to transmit malaria, but those whether that actually happens in the actual natural setting hasn't been demonstrated, but certainly in lab studies. So yes, we certainly have the vectors here, as do a lot of other states. We're not the only ones with that, but the species covers a lot of the eastern U.S.
Yeah, thanks, Jeff.
Thank you very much. We're going to keep moving right on down the list here to number 3, NPHL client services issues this month. I don't think there's much to report on from our end. I don't know if we have.
This is Brian Lenz. We don't have any issues to report that I'm aware of.
Okay, great. No news is good news. So we'll move into our outbreak updates. I know there's a lot of people on vacation this year. We'll try to see who's with us. For the HAIAR, I think Ishrat's on vacation.
This is Lacey. Yeah, her and Dr. Ashroff are both off today. I don't have any updates though that I can think of for the group.
Okay, thanks, Lacey.
Yeah.
Is Samir on for? Does not look like it.
He's
Okay.
out today.
Okay, so we'll do RSV and flu. Derek?
Yep, I'm here. So not a whole lot to report out for flu or RSV. Activity is still low as we expect during the summer months. Just a reminder, I think I've mentioned this before, but we are updating our influenza and other respiratory disease report on our website monthly over the summer months. We just updated it at the end of last week, so you should be able to view it on our website if you check it out today. Just a reminder, because it is summer, therefore agricultural fairs are going to be starting popping up, if not already. So if you do see any influenza A positives, especially a flu A unsubtypable, please let us know and get those to the public health lab. And that's really all I can think of. Robin, am I missing anything that we else need to report out?
Nope.
Alrighty, thanks.
Brianna, I think I saw Brianna was on.
Yeah, hi. We're talking about outbreaks. Is that right?
Yep.
I'm sorry, I joined.
Outbreak. Sorry, somebody was paying attention.
We're looking into a salmonella outbreak in the public health solutions jurisdiction, with just a family who prepared ice cream, like a homemade ice cream, and did not cook it, and it was made with raw eggs or farm fresh eggs. So we have a few individuals that tested positive for salmonella. So we're working on collecting leftover ice cream and eggs to see if it will be a match, and that was the cause of illness. So well, and some of the people, they got tested out of state. So we're working with out of state partners to make sure that those salmonella specimens get sent to their public health lab for whole genome sequencing. And then we have one here in Nebraska. So we've been working with that lab and notifying MPHL that might be coming their way here very soon. So what else?
Brianna, any more with the cyclosporas? I know we're seeing spread cases popping up.
Yeah, I was just looking at that. I was pulling that up right now. We are starting to see them pick up here in Nebraska, but right now for the whole year, we have 13 cases, which I mean, is still rather low, but it's picked up here. We've added about 6 cases in the last week or two.
Yeah, it's a little earlier because most of them usually show up mid to later July, it seems like. So
And
wonder what's
most
going on.
Yeah, a lot of, for what information I have right now, a lot of out of or international travel. And according to CDC, they don't include those in their national case counts. So they like to focus on United States exposures and what are people doing in the United States and eating in the United States. So I do know they're investigating a cluster of cyclospora. We don't have any cases a part of that yet. I was trying to look on I'm sorry, I feel unprepared right now. FDA's website where they post outbreak investigation tables where you can see what they're actively working on.
We sure want the laboratory partners to send us those positive stools that are cyclospora positive, because we are working with the CDC to do some other molecular studies on those. That's pretty important. And we do appreciate everybody sending us their positive cryptosporidium stools as well. We're part of a CryptoNet program for the US. I think we've seen a fair number of positive cryptosporidium too as well, Brianna, is that correct?
Yeah, yep. And typically in the summer months, we start seeing those. A lot of people are cattle branding and being out with animals and not washing their hands. So yes.
Yes.
I did, thanks for that segue, Pete, why I was able to pull up more information. But on FDA's website, they are investigating 2 cyclospora outbreaks. There's one cluster that has 37 cases identified in June, and then one just a week ago actually consisting of 105 cases. Both do not have products identified yet, but they are actively investigating. So yeah, as Pete mentioned, please submit your cyclospora positives to the public health lab. That helps with the national outbreak investigations. That's it.
Thanks, Brandon. Okay, next up, arbovirus with Jeff. Is there anything more?
Just a couple of things to add on. So, it is West Nile season. We've had some positive mosquito pools already, kind of distributed all across the state, not necessarily just concentrating in one area. We do have our one West Nile virus case. However, I was informed today by one of our local health departments out in the western part of the state that they had a patient who is hospitalized with meningitis that had a lab evidence of West Nile infection. However, they are not a Nebraska patient, but were likely exposed here. But since they're an out-of-state resident, they will not count as our case, but they likely acquired the infection here within the state. So West Nile virus is circulating around, and we're kind of starting to now see, our human cases pop up, it looks like, which is kind of what we tend to see. We see the mosquito positives come in, and then not too long after that, that's when we start seeing our human cases follow after that. So definitely West Nile virus circulating around. And then real quick on the tick side, we've actually seen a couple positive ehrlichiosis labs come in here just over the weekend. So certainly tick-borne diseases are still, ticks are still out and about as well. We tend to kind of see their activity decrease here as we get later into the season, but just kind of keep in mind that ticks are still active. And then, like I said, the West Nile virus is starting to pick up. So that's all I got.
Thanks, Jeff. TB testing, Kristen.
I don't see her on, Ben.
Moving.
I'm looking here.
Dana here for the STD program. Doesn't look like it. Okay, so we'll go on to lead testing.
Kristen said that she's having audio problems, but she had no updates. So there you go.
Good, I missed that.
No updates from the lead program today.
Okay. Thanks. I don't think there's too much to be said about agents tested at NPHL. We've been getting some things from Kansas, but that's usually Emily's jurisdiction. Pete, did you have something?
Well, I'm just going to say that we're helping Kansas State, the state of Kansas. We're helping Oklahoma, state of Oklahoma. We've done some help for South Dakota. So we're kind of almost become a regional testing site for this confirmation testing of these biothreat agents. And last week, we had a positive Francisella tularensis from Oklahoma. They tend to have a higher number than us, but I'm just saying, as we're seeing probably one isolate a week coming in that we have to confirm for these types of organisms. So they're out there. It's tularemia season, I guess. So be prepared in the lab if you think you might have one, because it potentially could be out there. Go ahead, Ben.
I was just moving on.
Okay.
Sorry to cut you off.
That's okay.
So LRNC, that's usually Emily again. I don't think there's been too much to update there. So we'll leave the laboratorians. We are going to be sending out the NPHL gram stain challenge next month. There'll be more information as we start to put that together in August. And then our wet workshop in North Platte is scheduled for September 14th. If you're west of Grand Island, expect an invitation to that today or tomorrow. Hoping to get our PACE credits put together, and then we're going to do it the next week after that in Omaha for the labs that are closer east of Grand Island. So we'll be sending out those invitations, probably directly to, lab managers. So keep your eyes out for that. Next meeting, Monday, August 14th at noon, we'll have another little educational thing put together for you. So for laboratorians, that's everything. Thanks for joining us. Again, feel free to get a hold of me or Becky with any questions or concerns, or if you just want to chat.